Cartilage and bone, skeletogenic tissues. Main questions of the lecture. Cartilage consists of cells, stem, chondrogenic cells, hondroblasts, presented with this electronogram, and older cells, chondrocytes, and big amount of intercellular substance consists of ground substance and fibers. No blood vessels inside cartilage. It's feeding by diffusion from perichondrium. There are three types of cartilage in the human body: hyaline, elastic cartilage, and fibrocartilage. Hyaline cartilage is transparent. Its name derived from hyalos, glass, so it looks like white glass. There is perichondrium around. It contains stem cells and hondroblasts, which can proliferate, and it provides the positional growth and regeneration of cartilage. Then zone of young cartilage consists of young chondrocytes, which continue to proliferate and produce intercellular matrix and embedded themselves inside, but continue to proliferate, dividing methodically, and from one cells can produce up to 8 cells. This group of cells called isogenic group, and division of mature chondrocytes provides the interstitial growth of cartilage, growth from inside. Hyaline cartilage located in trachea and bronchi, cartilage portion of ribs, articular cartilage, as well as in embryogenesis, the cartilage model of skeleton. This picture demonstrates the articular cartilage, which covers the articular surface of bones. It provides smooth surface between bones in joints. They work like shock absorber. It has no perichondrium, and feeding provide from synovial fluid and bone blood vessels. Elastic cartilage has a similar plan of structure as hyaline. There is perichondrium outside, contains stem cell and hondroblasts, which proliferate and participate in growth, peripheral growth of cartilage, as well as in regeneration, renovation after defects. Then zone of young cartilage and zone of mature cartilage with isogenic groups, which consist of 2-3 cells and located in right angle, perpendicular to the surface. This structure is like in hyaline cartilage, but in intercellular matrix situated the elastic fibers instead of collagen. This is the reason why elastic cartilage, the properties like rubber. It's really elastic. It's located in auricle of the ear, in epiglottis of larynx. fibrocartilage located in intervertebral discs in between tendons and bones make menisci of the knee joint. It consists of chains of chondrocytes with bundles of collagen fibers between. In embryogenesis, cartilage is developed from mesenchyme of sclerotom somites. So development of cartilage in embryogenesis from mesenchyme of somites of sclerotoms. Mesenchyme cells proliferate, produce a chondrogenic islet, and convert to hondroblasts, which produce intercellular matrix and embedded themselves, but continue to dividing, and under capsule isogenic groups of cells appeared. Bone tissue compose the bones. Functions: supporting the body, build the skeleton, protective, protect the brain, lungs, heart, other vital organs, and bone marrow from damages. Store of calcium, 99% of its content in the body located. Bone consists of cells, osteoprogenitor stem cells. They differentiate to osteoblasts, then to osteocytes. There are also osteoclasts, symblasts, under cellular structures. Intercellular matrix consists of collagen fibers and ground substance. 72% of minerals in bone. Minerals, salts, consist of calcium and phosphorus salts, and they make amorphous and hydroxyapatite crystals, which located parallel to collagen fibers. Osteoblasts, like fibroblasts in loose connective tissue or hondroblasts in cartilage, produce intercellular matrix, both collagen for collagen fibers and proteoglycans and glucoproteins for ground substance. Osteoblasts, big cells, contain a lot of organelles: endoplasmic reticulum, mitochondria, an electron microphotogram. There is part of nucleus and cytoplasm. These many organelles, especially rough and loose endoplasmic reticulum, Golgi complex. Many vacuoles, this is the secret. Osteoblasts secrete the organic components of the matrix, and this osteomucoid is lucid for electrons. Truly looks lucid, but later it embedded but calcium salt and became dense for electrons and black at electron microphotograms. So osteoblasts produced ground substance and fibers and embedded themselves in matrix. Osteocytes cannot produce matrix anymore. They contain few organelles and small nucleus, condensed nucleus with heterochromatin. But osteocytes still connected with one another, these long processes. So osteocytes located in bone lacuna and its processes located in bone canaliculi, and osteocytes connected between these long processes located in bone canaliculi. Osteoclasts, the symplasms, derived from blood monocytes, which fuse together and produce big structures, contain a lot of nuclei, up to 50, and a big amount of cytoplasm. Osteoclasts can destroy the bone, stain in blue, and they come to the surface of bone and destroy it and produce lacunas. Osteoclast adheres to the surface of bone by its peripheral part called clear zone, and in the center there is a ruffled border where the osteoclasts produce a lot of folds, big folds, and through these part of osteoblasts release carbonic acids produced from carbon dioxide and water. These are quite weak acid, enough to dilute the calcium salts, and calcium released and enter the blood and blood capillaries to increase the level of calcium in blood. Then enzymes of lysosomes release acid hydrolases which digest the organic components of bone. There are two types of bone tissue: Reticulofibrous, where collagen fibers are going in all directions, available only between skull bones in sutures and between some tendons and bone. Second part is lamellar ball, consists of plates. In every plate the collagen fibers are going in one direction. Dentin and cement of tooth also belong to bone tissues, but will be studied in the section digestion system, oral cavity and teeth. This slide demonstrates the reticulofibrous bone. These osteocytes connected by their processes and ground substance between, and collagen fibers in matrix going in all three directions. Lamellar bone consists of dense packaged lamellas in every lamella. Collagen fibers going in parallel. In another lamellas also parallel, but in opposite or another direction. These are circular lamella around Gavrsov canal. And between lamellas there are numerous osteocytes connected with one another by along processes, and oxygen and nutrition from blood diffuse through these canaliculi to the distant parts of bone tissue. Lamellar bone consists of compact and spongy cancellous bone. Compact bone composes the diaphysis of long bone and also cover spongy bone by thin layer. Spongy bone mostly in dead bone and epiphysis of the long bone. Bone as an organ. This picture demonstrates a piece of diaphysis of the long bone. You see transverse section and longitudinal section, and inside is marrow cavity. So there is periosteum around the bone and endosteum lining the marrow cavity. There are 3 layers of lamellas. First, located under periosteum, its outer circumferential lamella. Under endosteum, there is a layer of inner circumferential lamella. In between, quite thick osteon layer consists of osteons. These interstitial lamellas between, they are remnants of old destroyed osteons, remnants of previous generation of osteons. This picture demonstrates also the blood circulation. And then arteries enter the bone from periosteum. They enter the bone through the perforative channel and continue into blood vessels inside central canals, osteons. And there are also anastomosis between blood vessels of different osteons. Oxygen and nutrition enter the bone with arterial blood, and it diffuses from blood vessel in the central canal of osteon to the distant part of bone. And in opposite direction, CO2 and metabolite diffuse into the venous blood. Osteon, or Haversif system, is the structural and functional unit of lamellar bone. It consists of central canal with blood vessels and nerve, and up to 20 concentric lamella around, with osteocytes in lacunas between. All osteocytes connected by their processes. Osteocytes bodies are located in lacunas and processes in bone canaliculi. This is a microphotogram long bone diaphysis. There is periosteum around, the layer of outer circumferential lamella. The osteon layer consists of osteons and inserted lamellas between. And at high magnification, and there are 2 osteons with central canals, blood vessels inside, in the middle, and circular lamellas around. And these osteocytes between, and the processes in canaliculi going readily. Bone development or osteogenesis. Development of bone in embryogenesis. There are two types of osteogenesis: direct or intramembranous from mesenchyme, and indirect endochondrial at the site of the hyaline cartilage. This picture demonstrates the intramembranous bone formation. You see the mesenchyme of embryo. Mesenchyme cells proliferate, convert to osteoblasts, which produce intercellular matrix and embedded themselves. The osteocytes connected by invisible processes. From outside, the new generations of mesenchyme cells converted to osteoblasts, which cover the surface of bone tubercles in one layer, and they produce matrix and embedded themselves and stay in the middle. So by this the tubercles grow. And there are also osteoclasts, which connect to the surface of tubercles and destroyed the bone, remodeled the bone. By this way, the Reticulofibrous primary bone developed, and later it replaced by lamellar bone. Indirect endochondrial osteogenesis on the place of hyaline cartilage models of skeleton. Initially, hyaline cartilage models of bones appeared. For example, this is a long bone model, this diaphysis and two epiphyses. Osteogenesis start the replacement of perichondrium, and osteoblasts start to produce bone matrix, which calcified, and feeding of cartilage stop, and the cartilage destroy and died, and blood vessels grow from periosteum. Monocytes come in here, fuse, and osteoclasts appeared, and they destroyed the cartilage, and osteoblasts produce bone on the place of eliminated cartilage. So this is endochondrial ossification. Osteoclasts destroy bone and produce the primary marrow cavity here. The bone slowly growing, and then the secondary points of ossification appeared in epiphysis. It grew later the bone in diaphysis and both epiphysis, and only metaphyseal plate of growth of hyaline cartilage still exists. Cartilage hondroblasts dividing, proliferate and grow, and replaced for bone. By this way, bone grew and became longer up to 20 years old, when both plates of hyaline cartilage are replaced for bone. When it happened, the bone stopped to grow. Endochondrial osteogenesis. This is a picture of developing bone. Epiphysis and diaphysis. Epiphysis contain hyaline cartilage surrounded by perichondrium. The zone of intact cartilage contain azygenic groups. Then zone of columnar cartilage, where hondroblasts proliferate. Then vesical cartilage, when hondroblasts swollen. Then zone of resorption cartilage. Then osteoclasts come in and destroy hyaline cartilage. And then osteoblasts build the bone on the place of eliminated cartilage, these bone tubercles. And osteoclasts also destroy the new developed bone, and primary bone marrow cavity appeared. And you see the bone collar at the periphery of diaphysis, periosteum around. Bone regeneration, for example, after fracture of long bone. Initially, osteoblasts of periosteum and endosteum proliferate intensively. Then newly formed primary bone appeared, growing, and to replace the bone defect. This is initially Reticulofibrous bone, and later it replaced for newly formed secondary laminar bone. And this is a healed fracture. Remodeling of bone during the life, when the physical activity of the human changed, tendons for bone also changed. From the site of force action, osteoclasts accumulated and destroyed the bone, and from opposite side, osteoblasts proliferate and build the new bone.